In this module, we will talk about inhibition in enzyme catalysis. The rate of a chemical reaction catalyzed by an enzyme is not solely influenced by its substrate. It can also be significantly modified by other species through a process known as enzyme inhibition. An enzyme inhibitor is a special type of molecule that inhibits an enzyme by binding to it. It means that an inhibitor decreases the enzyme's activity and reduces the rate of chemical reactions catalyzed by it. In enzyme catalysis, inhibition plays a critical role in regulating the rate of biochemical reactions and controlling various metabolic pathways. Enzyme inhibitors can be reversible or irreversible. Reversible inhibitors attach non-covalently and can spontaneously dissociate, allowing the enzyme to resume its function. In contrast, irreversible inhibitors forms a chemical bond with the enzyme, permanently changing enzyme's functionality. Enzymes inhibitors function by either binding to the enzyme's active site, directly competing with the substrate, or attaching to a different site on the enzyme, thereby altering its ability to catalyze reactions. The role of enzyme inhibitors is significant in cellular regulation. They are often specific to a single enzyme and control its activity. For example, enzymes in a metabolic pathway might be inhibited by molecules produced later in the pathway, providing a negative feedback mechanism to maintain cellular balance. Additionally, enzyme inhibitors are vital in medicine as many drugs are designed to inhibit specific enzymes involved in diseases. In competitive inhibition, the presence of the inhibitor changes how the enzyme interacts with its substrate. Competitive inhibitors are molecules that structurally resemble the enzyme's substrate. Both substrate and competitive inhibitors can occupy the enzyme's active site, but unlike the actual substrate, competitive inhibitors do not react. Their binding prevents the real substrate from attaching to the active site, effectively inhibiting the enzyme's activity. In competitive inhibition, the inhibitor simply occupies the active site, blocking the substrate's access without participating in the reaction itself. Competitive inhibition can be described using the following mechanism, which is essentially a modified Michaelis-Menten mechanism. It represents the binding of the inhibitor to the enzyme and its effect on the substrate binding. In these equations, E represents the enzyme, S is the substrate, ES is the enzyme-substrate complex, P is the product, I is the inhibitor, EI is the enzyme inhibitor complex. The first equation shows the normal pathway of the reaction of the enzyme with the substrate, while the second equation shows the reversible binding of the inhibitor to the enzyme. Note that we assume that the inhibitor does not bind to the ES complex and the substrate does not bind to the EI complex. How does competitive inhibition changes the rate of enzyme-catalyzed reactions in comparison to the non-inhibited case, as described by the standard Michaelis-Menten mechanism? To understand this, we consider the total enzyme concentration and how it is distributed among the free enzyme, the enzyme substrate complex and the enzyme inhibitor complex. The total enzyme concentration is given by the sum of the concentrations of these three forms. Similar to the derivation of the Michaelis-Menten equation, we assume a steady state for both enzyme substrate and enzyme inhibitor complexes. For the concentration of the enzyme substrate complex, we apply the steady state approximation and obtain this equation, as we have already established before. Here, Km is the Michaelis constant that quantifies the substrate's affinity for the enzyme. From this relationship, we can derive the concentration of the free enzyme in the presence of the substrate. We now focus on the enzyme inhibitor complex EI. The rate of formation and dissociation of this complex is given by this equation. Applying a steady-state approximation, we obtain the expression for the concentration of the enzyme inhibitor complex. Here, KIC is the dissociation constant for the enzyme inhibitor complex EI. For further derivations, we express the concentration of EI as a function of the concentration of ES. Integrating previous equations into the equation for the total enzyme concentration, we get the following equation. Rearranging to solve for the concentration of enzyme substrate complex ES, we obtain the following expression. Returning to our expression for the reaction rate V, we derive the modified equation for the reaction rate in the presence of a competitive inhibitor shown here. The last equation can be reformulated as shown here. In this equation, Vmax is the maximum reaction rate for the original non-inhibited reaction. Comparing our equation for the reaction rate in the presence of a competitive inhibitor to the corresponding expression for the uninhibited case, we observe a slight modification. It can be described by the introduction of a new apparent Michaelis constant in the presence of an inhibitor. The term inhibitor concentration over KIC quantifies the effect of the inhibitor on the enzyme's affinity for the substrate. Note that in the absence of the inhibitor, the apparent Michaelis constant reduces to the original Michaelis constant. Now we can express the reaction rate under competitive inhibition as Vmax times the substrate concentration divided by the apparent Michaelis constant plus the substrate concentration. In the presence of an inhibitor, the apparent Michaelis constant Km star is greater than the original Michaelis constant Km. This indicates a lower affinity of the enzyme to the substrate compared to the uninhibited case. This inhibition effect is more pronounced if the inhibitor concentration increases or if the inhibitor has a stronger affinity for the enzyme. The latter is indicated by a lower value of the dissociation constant for the enzyme inhibitor complex. Consequently, the enzyme's apparent affinity for the substrate decreases, leading to a reduced reaction rate. The effect of competitive inhibition is illustrated in this figure. While the maximum reaction rate does not change, a higher substrate concentration is required to reach the same reaction rate in the presence of an inhibitor. The effect of competitive inhibition can also be visualized using a Lineweaver-Burk plot represented by this equation. In this plot, because Km star is larger than Km, the slope of the line in the presence of an inhibitor is steeper compared to the slope in the absence of an inhibitor. In uncompetitive inhibition, the inhibitor binds exclusively to the enzyme-substrate complex, not to the free enzyme, and forms an enzyme-substrate inhibitor complex, denoted as ESI. This distinct binding mechanism leads to alterations in both the enzyme's maximum reaction rate and its affinity for the substrate. The reaction mechanism of the uncompetitive inhibition can be described by the following reaction scheme. In these equations, ESI represents the enzyme-substrate inhibitor complex. The first equation illustrates the normal reaction pathway of the enzyme with its substrate, while the second equation shows the reversible binding of the inhibitor to the enzyme-substrate complex. Applying a steady-state approximation for the enzyme-substrate inhibitor complex, we derive the dissociation constant Kiu for it as shown here. The reaction rate in the presence of an uncompetitive inhibitor is then given by this expression. This rate equation can be rewritten in terms of apparent parameters. The equations for apparent parameters of the Michaelis-Menten equation in case of uncompetitive inhibition are shown here. It is crucial to note that both the maximum reaction rate and the Michaelis constant decrease in the same proportions due to the uncompetitive inhibition. This effect is evident in a Lineweaver-Burk plot of uncompetitive enzyme inhibition, where the slope of the line, representing the ratio between apparent Km and Vmax, remains constant regardless of the presence of an inhibitor. Furthermore, the decrease of the maximum reaction rate in uncompetitive inhibition indicates that this type of inhibition cannot be overcome by increasing the substrate concentration. In non-competitive inhibition, the inhibitor binds to an allosteric site on the enzyme, which is different from the active site where the substrate binds. This binding changes the enzyme's structure in a way that reduces its activity, even if the substrate is bound. The key feature of non-competitive inhibition is that in the presence of a non-competitive inhibitor, the enzyme's catalytic efficiency is reduced by decreasing the turnover number, rate constant K2, while its affinity for the substrate, quantified by the Michaelis constant, remains unchanged. Unlike uncompetitive inhibitors, non-competitive inhibitors can bind to the enzyme whether or not the substrate has already been bound. Their binding reduces the overall number of active enzyme molecules available. This is because a portion of the enzyme is always inhibited from converting the substrate to the product, effectively reducing the concentration of active enzyme. The apparent enzyme concentration in the presence of a non-competitive inhibitor can be represented by this equation. Here, concentration of E0 star is the apparent total enzyme concentration. Concentration of E0 is the actual total enzyme concentration, and K in is the dissociation constant for the enzyme inhibitor complex in a non-competitive inhibition. KEN quantifies the affinity of the inhibitor for the enzyme. The reaction rate in the presence of a non-competitive inhibitor is given by this equation. It shows that the maximum reaction rate is reduced in the presence of the inhibitor, leading to a new apparent maximum reaction rate Vmax star. However, the Michaelis constant remains unchanged. This characteristic distinguishes non-competitive inhibition from other types of inhibition, as it affects the enzyme's catalytic efficiency without altering its affinity for the substrate. In mixed inhibition, the inhibitor binds to an allosteric site on the enzyme, which is a site different from where the substrate binds. This binding occurs whether the substrate is already bound to the enzyme or not. The key characteristic of mixed inhibition is that it affects both the enzyme's maximum reaction rate and its affinity for the substrate. When the inhibitor binds to the enzyme-substrate complex, it reduces the enzyme's catalytic efficiency, similar to uncompetitive inhibition. However, when the inhibitor binds to the free enzyme, it also affects the enzyme's ability to bind the substrate, similar to competitive inhibition. This dual nature of mixed inhibition results in a decrease in the maximum reaction rate and a change in the Michaelis constant, which can either increase or decrease depending on the specific interaction between the enzyme, substrate, and inhibitor. The Lineweaver-Burk plot, a graphical representation of enzyme kinetics, is particularly useful for distinguishing between different types of enzyme inhibition: competitive, uncompetitive, non-competitive, and mixed inhibition. Each type of inhibition has a unique effect on the plot, allowing for clear differentiation. In competitive inhibition, the inhibitor competes with the substrate for the enzyme's active site. On a Lineweaver-Burk plot, this results in an increase in the apparent Km while the Vmax remains unchanged. Graphically, this is seen as lines with different slopes intersecting at the same point on the y-axis, which represents 1 over Vmax. Uncompetitive inhibitors bind only to the enzyme-substrate complex. This type of inhibition decreases both the apparent Km and Vmax in the same proportion. On the Lineweaver-Burk plot, this is represented by lines that are parallel to each other, indicating a constant ratio of Vmax over Km. Non-competitive inhibitors bind to an allosteric site on the enzyme, affecting its activity regardless of whether the substrate has bound. This leads to a decrease in Vmax but does not change Km. On the Lineweaver-Burk plot, this results in lines with the same x-intercept, representing the negative of 1 over Km, but different y-intercepts. Mixed inhibition is a combination of a competitive and uncompetitive inhibition. The inhibitor can bind to both the enzyme and the enzyme substrate complex but with different affinities. This results in changes in both Vmax and Km. On a Lineweaver-Burk plot, mixed inhibition produces lines that intersect at a point that is not on the axis, indicating changes in both parameters.